Dr. Bokara Kiran Kumar, Senior Scientist working with CHIR CCMB. We cordially invite you for this webinar, sir. Thank you so much for accepting our invitation. Welcome to the webinar, sir.
Thank you.
Thank you, sir. Now, I would like to call upon my colleague, Dr. S. Swati, to invite other guests also to this webinar. Swati ma'am, over to you.
Thank you, Shravanti. Good afternoon, everyone. It's a great opportunity to invite dignitaries to the webinar. So now I am glad to welcome Chief Patron of this webinar, Dr. R.S. Praveen Kumar, IPS, Secretary TSW REIS and TDW REIS. So welcome to the webinar, sir.
Stem cells webinar.
Prasanna, please unmute the participants, please. I would like to invite
Madam, mute on the madam.
You are in mute, ma'am. Unmute yourself.
I'm on unmute only. Sorry for the disturbance. I would like to invite patron of this webinar, Dr. Praveen Mamidala, Joint Secretary, Head of Higher Education Department, TSW REIS. Welcome, sir. Now I would like to invite Coordinator, Dr. P. Banu Prasad, OST, TSW RDCs. Welcome, sir. It's my honor to invite Chief Advisor, Dr. P. Rajini, Zonal Coordinator, South Region. Welcome, ma'am. Now I invite Convener, A. Himabindu, our beloved Principal, TSW RDCW Jagadgiri Gutta. Welcome, ma'am. And I also invite Co-Convener V. Nutana, our Vice Principal, TSW RDCW Jagadgiri Gutta. Welcome, ma'am. And now I welcome all my colleagues, faculty members, research scholars, my dear students, and all the participants to the webinar. So now I invite Zonal Coordinator, Dr. P. Rajini, ma'am, to address the gathering. Over to Rajini, ma'am.
Okay, thank you very much, Swati. To start with, I want to congratulate the Department of Zoology, Jagadgiri Gutta, for conducting such a good webinar on the stem cells, hype or hope. Very good topic I have selected. This is the future, I believe, in medicine. This will be changing a lot. So the scenario will be completely changing when research is better done on these cells, stem cells. And I also congratulate and then thankful to be Dr. B. Kiran Kumar Garu from CCMB for accepting the invitation and spending time for our students in enlightening on stem cells. There's not much to say, and I also congratulate all the students who are present to this webinar, and I hope all the students know something about the stem cells. They are using the stem cell. A word itself is has become a passion nowadays. So how best we can use it, whether it can be used for the betterment or for the worst. That is there in the hands of the human beings. And I want to tell the students that these are very special human cells. Stem cells are very important, special cells, which may be developed into many different types of cells, starting from muscle cells and brain cells also. Therefore, you can feel the importance of this stem cell. Sometimes they may fix up the tissues. So in many cases, they have done much, but still stem cell research must go on. It is not so much, I do not know. Due to some failures, I think they have stopped. A few of the countries, they have stopped. And I think our students, who are the future of the world, must put in your mind towards this. And then I request you all to do some sort of a research which will change the world, which will make a difference in the present. please, I request you all to take up. And this is a very good this thing. And definitely, stem cells are hope according to me. Hype is nothing but the media has given a hype. That's all I feel. Stem cells are good. The way we use it is the main thing. So topic is very good, and I request all the students to listen to it carefully and note down the things and submit to your department head. Thank you for giving me this opportunity. Now I request the next speaker to take up the mic. Thank you. Thank you very much, Swati.
Thank you, ma'am. Thank you for inspiring us always. We are always very much interesting to listen interesting words from you, ma'am. Thank you, ma'am.
Thank you.
Now, I would like to invite our principal, Himabindu ma'am, to address the webinar. Over to Himabindu ma'am.
Good afternoon, ZCO ma'am. Good afternoon, ZCO, sir, and all staff and the students. First of all, I would like to thank Swati and Shravanti for organizing a webinar on such a wonderful topic. And I thank our gracious ZCO madam for accepting to be our guest for the webinar, and they were encouraging. And then I welcome our chief guest speaker, Dr. B. Kiran Kumar Garu, senior scientist in CSIR and CCMB. Sir has graciously accepted our invitation to be the chief resource person guest speaker for today's webinar. Sir, I like to thank you for this opportunity given to our students to hear a wonderful informative lecture from you on stem cells, which is whether it is a boon to the medical field or there are some ethical issues related to that. We'll know from you, sir. And this webinar is one way of keeping in touch with the academics for the students in this time of pandemic. And such webinars help our students to keep on gaining knowledge. And sir, I welcome you to this program, sir. Thank you, Swati.
Thank you, ma'am, for addressing the webinar. Thank you so much. Now, I would like to call upon B. Shravanti, ma'am, to welcome today's resource person, none other than Dr. B. Kiran Kumar, sir. Shravanti, take over the mic.
Thank you, ma'am. So I'm considering it as a great privilege to introduce today's guest speaker, Dr. Bokara Kiran Kumar, senior scientist, CSIR CCMB, to the webinar. Thank you so much, sir. Sir has completed his PhD from the Department of Zoology, Andhra University, Vizag, in the year of 2007, with the topic Studies on the Toxic Impact of Sublethal Lead in Posterior Post-Larvae of Penis Monodon, under the guidance of Professor Milton Fingerman, Tulane University, USA. Sir started his research career in the year of 2001. Sir has worked as a research mentor, junior research fellow, senior research fellow, research associate, research professor, and postdoctoral research fellow. And sir currently working as a senior scientist in the CSIR CCMB. Now sir also handling with 6 projects. So far, sir has completed 4 MIRT projects and 2 MHIRT projects and 1 project in the Neuroscience Laboratory, Department of Anatomy, Seoul, South Korea. And the topic is characterization of transgenic neural system, stem cells and their applications for controlling neuronal diseases both in vitro and in vivo. Now, sir handling with 6 projects in the area of stem cells and SARS-CoV-2 also. Sir has collaborated with reputed universities and institutions like LV Prasad Eye Institute, IITH Hyderabad, NIN Hyderabad, Alcorn State University, Lorman, MS under the MHIRT funded by National Institute of Health, USA. Sir has attended many workshops, seminars, conferences, symposiums, and sir presented number of posters also. Sir is a life member in Society of Neurochemistry, India, and he is reviewer in reputed journals. He is the stem cell expert member for the Institutional Committee for Stem Cell Research, NIT Warangal. Sir worked as a co-investigator for 3 CSIR projects, working as principal investigator for DBT project also. He guided so many students, projects, currently guiding 4 JRF students and 1 PDF student also. Sir published more than 25 research articles in international journals and number of articles in national journals also. He contributed his work in the book entitled Stem Cell and Cancer Stem Cells, the book entitled by Spingerman, Volume 7. Sir delivered many lectures across the nation, and it is very short description about Sir addressing. I took it as a great opportunity to introduce Sir. Thank you so much, Sir. Now I'm handovering the session to you, Sir. Sir, take over the session. Thank you so much.
Yeah. Thank you so much for your kind introduction. It's been nice hearing the things which I don't know also. I guess the audience are undergraduates, and they might be having some kind of a background related to stem cells or basics of stem cells. So I can share the screen now, right?
Yes, sir. Okay, sir.
Okay, I think you have to stop the screen screen sharing.
I have stopped, sir.
Just stop it.
Are you able to share the screen, sir?
Yeah, I'm just trying it. So can you see it? Can you see the screen now?
Yes, sir.
Okay. Okay. Before we start the topic related to the stem cells, we would like to address the gathering, or can just pose a question to the gathering whether the stem cells, which are considered to be a hope, is it been hyped? So with this question, we'll start this session. It mostly relate. I kind of present my contents mostly like a discussion rather than a lecture part. Okay, so we'll discuss the things which are happening, or we'll discuss the things which are actually existing, and what is hyped and what is the hope. Okay, so the first slide, what you're seeing is 3 different cell types which have been differentiated from a single cell type. Okay, that itself indicates the stem cell has the potential to differentiate into different cell types. So I took mesenchymal stem cells from the dental pulp, and then you can see my cursor, right?
Yes, sir. Yes.
Okay.
Yes.
Yeah. So I could able to differentiate from mesenchymal stem cells into 3 different cell types, which have different functions. One is the adipocytes, which give rise to the fat cells. One is the osteocytes, which gives rise to the bone cells, and one for chondrocytes, which gives to the cartilage. Okay. So these 3 different cell types we have differentiated from a single cell type, indicating like a single cell, one cell type could be able to differentiate the different cell types. That indicates like they have a capacity to form more cell types, which is considered as one stem cell. That's what a stem cell is, a cell which harbors the factors which could differentiate into different cell types. This is one core example wherein we have differentiated the mesenchymal stem cells into 3 different cell types. One is adipocytes, osteocytes, and chondrocytes. Okay. Now, so I will navigate my topic with 3 different subtopics. One is what are stem cells in the clinic, and I would like to present some of our lab results or preclinical results wherein we have used stem cell therapy for some kind of pathological intervention. If you see on the slide, for the stem cells, you need to have a very good microscope to observe the cells. You need to have a very good cryogenic facility to, this is the cryogenic facility where there is a liquid nitrogen which goes to a temperature of -170 degrees centigrade, where you preserve the cells for the future use. And the green, you can see some green cells and you can see some blue cells here. Okay, the green represents the stem cells, the blue represents the normal cells. You photograph this slide, we will discuss more detailedly about what is green and what is blue in the coming slides. Okay, let us go for some little introduction about what are stem cells are. Of course, they should have a very good self-renewal and differentiation capacity. Self-renewal in the sense like one cell gives rise to an identical daughter cell, which will not go for differentiation. And they should have an ability to proliferate and without the loss of differentiation potential. Or you can say like, it should not go for senescence, means they should not age. Aging cells are non-stem cells, whereas stem cells will not age. I mean, like in the sense like they're considered as eternal. You can say like they have to have the potential to differentiate multiple types, multiple times without losing their telomeric ability. I mean, the DNA should not be damaged so that the difference, their proliferation capacity should be going on. Okay, and then as I said, like the stem cell is a very blank cell, means you can signature anything. A stem cell can give rise to any cell type depending on the origin. Either it may be, and it depends on the origin of the cell, whether it's like embryonic in nature or adult in nature. So depending on the nature of origin, the differentiation potential of that particular cell type varies. This we'll discuss more detailed in the coming slides. And essentially, every human being is being built from a single cell. That indicates the originating cell, which is at the time of fertilization, the ova and the sperm are the starting point, or the stem cells that are present in the germ layers would be able to give you the whole organism. So any organism starts with a single cell, and that single cell is the stem cell, which gives rise to multiple cell types. That you can say the body comprises of 200 different cell types. These 200 different cell types are being originated from a single cell type. Okay, so this is about the small introduction.
No.
First thing is like they should have a capacity to self-renewal and also differentiation, and they should continuously proliferate without losing the differentiation potential, without senescing also, means they should not age. And it should be a blank cell where it can be a signature to create multiple cell types, which can be like a fibroblast, that is skin cells or muscle cells or nerve cells or cardiac cells, whatever it is. It starts from a single cell type. So it is considered as the building block of human body, as like the amino acids considered as the building block of the proteins, whereas the whole structural organism or the body comprises from a single cell type, that is stem cells. Now the same thing, the features is like when these are different from other cell types because they have a controlled cell cycle mechanism, because every cell should obey the principle called as cell cycle. There should be a G1, G2 and S phases, wherein the cell goes in a different phase of their life. So these stem cells are very unique, wherein they will restrict or they will make themselves govern to the cell cycle mechanism. That's the reason why they have the ability to self-renewal capacity and also the differentiation potential. And as I said to you before, they, depending on the nature of the signals at the time of development, every single cell type will go for a different cell organelle, or you can say like It can go to a muscle cell, it can go to a nerve cell, it can go to a cardiac cell, it can go to a liver cell. So different cell types will give you will be originated from this. At the same time, stem cells are able to the self repair mechanism that is happening inside the body, because every time there will be wear and tear in the body, and this wear and tear will be taken care by the endogenous stem cells that is present inside the body. Okay, and stem cells are considered to be eternal, means in the sense like they have a potential to differentiate, or you can say like divide without limit in a living organism, so that they will replenish or they will replace the lost cells. So every time this mechanism always happens in the body, wherein the new cells come in and the old cells are thrown off. This happens because of the endogenous nature of the stem cells presence. And as I said you before, the stem cell divides, and each cell has the potential to either remain a stem cell or to become as an other type of cell type. Okay, as I said you, from the mesenchymal stem cells, we could able to produce the bone cells, you can be able to produce the cartilage cells, you can be able to produce the fat cells. The same way, from the stem cell, depending on the signal what the body requires, they could be able to transform that cell type to a specialized cell type which the body requires. For example, the muscle cell or the RBC cell or the brain cell or the cardiac cell, whatever it is, depending on the requirement which is required for the body. Now, what are the unique properties? Unique properties are, the first important thing is like the renewal capacity for the longer periods, and then to the beginning they are unspecialized. I can say like they are very blank. Depending on the requirement of the body, these unspecialized cells will become to a specialized cells to form a different cell type for the function. And as such, I said it before, previously they are unspecialized or they're uncommitted. Uncommitted means they're not committed for any kind of cell type until unless there is an internal signal that gives that particular stem cell to form into a different cell type. Okay, so a stem cell is uncommitted until it receives a signal to develop a specialized cell. So this makes unique demarcation on the cell type, basing on the stem cells or the non-stem cell. Now, I have said it before, stem cells as such can be divided in different functions, and depending on the nature of the differentiation, they are classified into different terms. So as I said, the classification of stem cells will be based on the source of origin, at the same time, the action potential. So we have to clearly understand this terminology before we venture into the aspects of stem cell function. So first of all, source. The source may be either embryonic or adult. So embryonic state means they're considered as at the birth. Like when you say about embryonic, we consider as the umbilical cord or extra embryonic tissues, which could be a source of the stem cells. When I consider as adult, even the mesenchymal stem cells that are present in the adults, or you can say like the fully grown organism, could be a source of the stem cells, because when during the developmental process, everywhere there will be wear and tear, and everywhere there is an aging process happens inside the body. And during the aging, the old cells goes off and the new cells come in. How the new cells come in is because of the resident stem cells that are present inside the body, and this potential of producing new cells for the whole life will be kind of compromised by aging. So that's the reason why at the aging, or you can say like at the older ages, the repair of the body is less or compromised compared to the juvenile stage or childhood stage or young stage. Coming into the potentiality of the stem cells to form different cell types, the terminology can be different things. One is the totipotent nature, pluripotent nature, multipotent nature, and unipotent nature. The best examples, or you can say like the dissection of this terminology into the action is the totipotency means when a cell type could able to produce a full organism. I repeat it again, when a cell type a single cell type producing a full organism, then those particular cell types are considered as totipotent cell types. The best examples are like the germ cells. The germ cells which are like sperms or the ova, they are considered as totipotent because those two single cell types could able to produce the whole organism. Coming into the pluripotent cell types. Pluripotent cell types is something like they can produce the organ systems, not organism itself. So a cell type which could able to produce an organ system, not organism. Cardiac cells. So the cardiac producing cells are considered as or one single cell type which can produce the muscle cells, the bone cells, the heart cells, the liver cells, or the nerve cells. These kind of stem cells are considered as pluripotent stem cells. Okay, they could produce an organ system, whereas the multipotency have some restriction in the lineages. For example, if it is mesenchymal stem cells, the mesenchymal stem cells we consider as multipotent rather than the pluripotent. Why? Because these stem cells are very confined to specific differentiation, wherein they cannot form all cell types, but they're limited to certain cell types. Okay, unipotent in the sense they produce only one single cell type. For example, hematopoietic stem cells. Hematopoietic stem cells will produce only blood cells, but they cannot produce the other than the blood cells. So those are considered as unipotent. So depending on the nature or action of, or you can say like the potential of the cell, the ability or the action potential varies, and the terminology is termed differently for their actions. Okay, so now you can see a small diagram which actually shows the developmental continuum from a single cell embryo. You can there will be a 3-day embryo. From 3-day embryo, there will be a ICM called as intercellular mass, which comes at 5 to 7 days embryo. These are the source of embryonic stem cells. That's what we say, like after the fertilization, after they form some divisions, and the division after 5 to 7 days, the mass that is present at the intercellular level, that's intercellular mass, ICM, is considered to be the source of embryonic stem cells, which are considered as totipotent, wherein they have the potency to form the full organism. Okay, and when it having the developmental continuum after 4 weeks to 6 weeks, they are having the ability to form specific organelles, or you can say like specific organ systems. That's why we consider as pluripotent in nature. So totipotency, they form the full organism, whereas the pluripotency, they can form the organ system, where they can form the limbs, they can form the legs, they can form the brain, they can form, but they cannot form the whole body. Okay, so another thing is like the fetal stem cells, which may be a pluripotent or multipotent. The fetal tissues, that is extra embryonic tissues, which are actually derived from the fetal tissue. And there is a lot of interest going on cord blood preservation for future use of the stem cells for any ailments. That's called as cord blood cells or placental stem cells. They are also considered to be pluripotent, or sometimes they are multipotent, depending on how you are harvesting the tissue. That governs whether you are having the pluripotent nature of the cells or you have the multipotent nature of the cells. And later on, we consider there is another thing called as adult stem cells. The adult stem cells may be the pluripotent in nature or multipotent in nature. And the embryonal carcinoma cells, the carcinoma is also have the stem cells. They are also pluripotent in nature. So these are the various stages from the life wherein the source of the cells and the function of the cells varies depending on the developmental nature of the organism. At the earlier stage, the cells are totipotent in nature. Later stage, the cells may be pluripotent or multipotent in nature. Yeah, let us dissect each one into more detail. The totipotency have the capacity to produce all cell types of the developing organism. It includes both the embryonic and extraembryonic tissues, example placenta. Whereas the pluripotency, they can only make cells of embryo proper, but make all cells of the embryo, including germ cells. Therefore, so the pluripotent nature of the stem cells have an ability to produce ecto, endo, and mesodermal layers. That's called as germ layers. The germ layers producing cell types are considered the pluripotent stem cells. Whereas multipotent stem cells, they are within the germ layer. So they are actually extracted from the mesodermal tissues like blood, which can make cells of the blood, but they cannot make another cell types of ectoderm. So when a multipotent cell type has been taken, that multipotent cell types may be either from ecto, which gives rise to maybe neural tissues, mesoderm, which gives rise to the blood cells, endoderm, which gives rise to the gut and other complex tissues. Okay, and unipotent stem cells, they are only single cell types. They cannot produce. Example is a germ line, germ cell type that makes the cells that mature to become egg or sperm, but not other cell types. So a single cell type is unipotent, but the egg and sperm are mixed together, then they become as totipotent in nature. Okay, now the first slide which I showed you had green, but the green is replaced by red here, and the blue remains as blue. Okay, when you consider, okay, what is the unique features wherein you identify stem cell to a non-stem cell? Okay, now there are different markers to establish the signatures or to identify the nature of the cell depending on the surface markers, or you can say like cluster of differentiation markers, CD markers. Okay, so these CD markers are the signatures to identify whether this is a stem cell or a non-stem cell. There are different signature markers to identify them. So one is like CD105. So this CD105 is one of the stemness marker, which actually showed in red, and whereas the other cell types are shown in blue. Now, pluripotent stem cells indicates like there are more number of stem cells which can give rise to more different cell types. So the tissue stem cells, you can see there is only very small portions of the red population, indicating like in the tissue, the stem cells are very limited. At the tissue level, the stem cells are very limited. Whereas when it comes to that embryonic state, the stem cells are more in number. That indicates like during the developmental process, the population of the resident stem cells keep on compromising because of the continuous proliferation nature that happens inside the body for taking care of wear and tear of the body. And I have to other things like the tissue specific stem cells, or they're considered as the adult stem cells. These are either isolated from the fetal tissues or the umbilical cord cells, umbilical cord blood. The most difficult part is because they are very, very finicky in nature. It's very difficult to isolate, and it's also difficult to grow more in the laboratory also. And mostly they are multipotent in nature, and they also involved in the tissue repair and also the homeostasis, which should maintain the balance of the cells that goes off during the aging process. So I can say like these are the different markers which you can identify. The hematopoietic stem cells should be CD34 positive, whereas the mesenchymal stem cells should be CD34 negative. These are the cluster of differentiation, that CD represents cluster of differentiation markers wherein you can identify whether this cell type is kind of a specific cell type or not. And the other one is embryonic nature. So embryonic stem cells, as I said before, they are originated from the embryo at the stage of 5 to 6 days after the fertilization. And the mass you can see here is the intracellular mass, which are actually the source of the stem cells. So these particular cell types have the expression of the markers representing the stem cells are high in number. That's why they have the highest potential to proliferate in the laboratory condition without going for differentiation. And they retain the ability to form different tissue types. And that's the reason why we consider this as the pluripotent stem cells. Okay, now, as I said you before, that isolating the embryonic states, cells from embryonic state has attracted most lot of ethical concerns, because you need to abort the fetus to actually take the ICM, intracellular mass, to go for totipotency or pluripotent in nature. But in the this early 20th century, tremendous work done by the Robinson and then Yamanaka, wherein they have created a platform to actually replace the embryonic stem cells which are pluripotent in nature, to isolating the pluripotent nature of the cells from the adult tissues. Okay, so I repeat it again: the embryonic nature of the stem cells are considered to be pluripotent, and it attracts a lot of ethical issues because they are originated from the IVFs or aborted fetus. So the work which has been created by Robinson and the Yamanaka has developed some kind of what you can say, like identifying the crucial transcriptional factors, wherein these transcription factors could able to govern to form whether to form the cell to be pluripotent in nature from the adult cells. I'll explain in more detail in here. So these IPSCs are generated by using these 4 transcriptional factors called as c-MYC, OCT4, KLF4, and SOX2. These are the 4 transcriptional factors which are essential for a cell to make them pluripotency. So if you could able to induce these 4 factors in any cell type, either it may be adult cell or it may be normal skin cell or adult cell, if you are able to introduce these 4 transcription factors, that particular cell type from the adult nature, it comes back to the embryonic state, wherein it can be functional as pluripotent in nature. It's a very great discovery that that has been done in that 2002, 2012, like 20 to 20 years from now, 18 years. So this particular discovery has led them to grab a Nobel Prize, and then this particular methodology is now employed to actually create embryonic-like stem cells from the adult cells for regenerative medicine. Okay, so what could be the advantages of this? Advantages like, why do you need this kind of that's why they consider as induced pluripotency. You have induced the pluripotency in an adult cell to make it pluripotent. That's why the name termed as induced pluripotency. So they can create stem cells directly from the patient. If the patient has a problem, then if they need to have some kind of a organ therapy, then you can actually take the cells from the patient, and then you can make the patient cells to form embryonic in nature, and then you can differentiate those cells to the specific cell type which the patient requires for the transplantation. Okay, and as I said you, this pluripotent nature of the cells could able to maintain indefinitely in the laboratory without differentiation, and they share equal properties. They share equal properties with embryonic state stem cells. So you are actually eliminating the ethical issues. You are eliminating the cumbersome process of isolating the cells from the IVFs. You are just introducing some transcriptional factors which regulates the stem cells to form induced pluripotency. This small nature or this small tweaking of making the cells has given a hope for most of the therapeutic applications. Now, this particular checkerboard shows you like, yeah, this is the, you can, from the patient, you can take the skin. These skin cells could, we can actually induce these 4 transcriptional factors, and then these particular cells will become as patient specific IPS cells, or as these particular cells are derived from the patient itself. And how, what could be the advantage of having these things? There are 2 different advantages here. Here, you can use these cells for gene targeting, causing genes. If you have any kind of mutations that is giving disease to the patient, that particular cells which are taken from the patient can be repaired through CRISPR-Cas9 mechanism, and then you can correct the defective genes, and then you can make these cells healthier, and then you can transplant these genetically modified cells to the healthy to the patient itself, so that these particular cell types which are having that genes which can correct that disorder can be integrated inside the body, and they can make, and they can do the repair mechanism. Okay, and other type is like this particular patient specific iPSC cells, you can make the in vitro differentiation, and this differentiation could able to use for screening the therapeutic molecules, not onto the patient directly, but onto the patient cells, so that this could be a personalized medicine for him to take the drugs. So before you recruit the patient, you can take the patient cells and then see the response of the drug, and then you can recommend the drugs to the patient. So these 2 kind of advantages lies with this iPSC cells. Okay, what could be the difference between these 2? The source is embryonic stem cell source is embryo, whereas iPSC source is somatic cell. That's what I said. Like from the skin you can take and you can go for differentiation. And the tissue specific cells, there can be either adult cells, they can be juvenile and fetal organs. However, I said you before, the proliferation rate in ASCs are very high, and iPSCs, because they kind of share the same properties of ESCs, their proliferation is also higher. Whereas the tissue specific cells, they are very low. That was, I have shown you before one slide, wherein in the tissue, the number of stem cells presence is very limited. Availability is very high, and the tissue specific cells are very low. The differentiation, spontaneous differentiation is very high in ASCs and iPSCs, but it is very low in tissue cells. And the capacity to produce the diverse cell types are higher in ASCs and iPSCs, whereas it is very low in tissue specific stem cells. Okay, why the stem cells make so valuable? Why you want to have the cells to be studied so well and understand the biology so well? Because they are the future for the therapeutic applications. When you consider biology, the pluripotent stem cells or the tissue specific stem cells, this nature could able to produce different cell types from a single cell type. It can produce the hematopoietic stem cells, it can produce endothelial cells, it can produce muscle cells, it can produce neuronal cells. And the applications orienting from this biology is, you can use it for cell therapy, wherein you can transplant specific cells and specific precursor cells for different tissue types. And you can do a very good research on it, wherein you can see the genetic or molecular changes that happens during the tissue growth or during development. And also you could able to understand the functions of the genes or the proteins that are actually crucial for the stemness to retain the stemness in the coming days. And the new drugs can be screened by using this this information from the stem cells, so it can be used for the drug and chemical development as well. As the name indicates, like, no one cell type fits all applications, but and you need to do the research to check the stem cells from different cell types also. So this could be one particular area that has to be seriously thought of, and people are working more on understanding cell types and also the resident stem cells in the specific organ niches. Okay, with the small introduction of the stem cells and their biology, or you can say like the nature of the stem cell, or you can say like the differentiation ability of the stem cells, I would like to navigate you to some of my lab findings, wherein I use stem cells as uh, one of the application to actually repair, or you can say like check for therapeutic applications. The first head to understand how the cell therapy could able to address the spinal cord injury. Well, as everybody knows, the spinal cord injury is a very drastic, or you can say this uh, very not good ailment wherein there will be a permanent disability or loss of movement. It may be either paraplegia or it can be a tetraplegia. When paraplegia, your forelimbs or hind limbs, any one of them are compromised. Wherein in tetraplegia, the both forelimbs and hind limbs are paralyzed, and there will be a compromise of the quality of life. So uh, most of the spinal cord injury happens because of the accidents, sudden accidents, and this will compromise the quality of life of the patient. So depending on the nature of the injury, either it may be at cervical region or it may be at thoracic region, the ailment differs for different people. So you can see this is the spinal cord here, and this is being innervated by the nerves endings, and the discs and the vertebrae are the cushioning pads wherein it, it maintains the compactness of the spine. And this is the cross section of it. And then mostly by the accidents, there will be a kind of compromisation on these ligaments or joints, so that the organs which are associated with the spinal cord will be compromised. So we can consider as the when you take the spinal cord, it has been divided into 4 different regions: the cervical, thoracic, lumbar, and sacrum or coax. Most of the accident happens in the thoracic region, and sometimes it can be at the thoracic cervical region. So when if there is a injury at the cervical region, that is at C5 to C4 to C6 segments, it leads to complete paralysis of both forelimbs and hind limbs. That condition we term it as quadriplegia. If it hits at the thoracic region, the hind limbs will be compromised, and the person not able to walk, and that particular condition is called as paraplegia. The same thing happens with the lumbar region. So spinal injury is very common, and it could be addressed more efficiently with these cell therapy mechanisms. We have published a paper way back in 2000s, wherein we have shown that the agmatine is one of the molecule which is a neuromodulator, could able to help the recovery of the spinal cord injury after the treatment. So these are some of the findings here. I don't want to go details about this paper, but I'll tell you what are the strategies that we kind of adopted in our in vitro in vivo model in CCMB. One thing is like when injury occurs, injury occurs, it will have 2 different events called as the primary events and the secondary events. The primary events is of axonal degeneration, demyelination, and the loss of function, and secondary events leads to ischemia, inflammation, and vascular changes. The primary event, if it is not attended, it leads to the secondary events, which ultimately leads to the compromise of the locomotion. If you see here, what is the treatment strategies that are available for spinal cord injury? It may be like the neurotrophic factors, which could able to stimulate the axonal growth, and it should have some kind of ability to replace the injured cells. But as of now, the existing treatments are that normally they go for therapies, or they go for surgeries, or they go for a high dose of steroid injections, and these steroid injections have some other secondary complications, and they might reduce the inflammation, but they are compounded with the secondary complications. And these are various complications that is associated with the existing treatments. It is like there's a deterioration of the neurons, and there's a lot of pressure sources, and there is a problem with the breathing issues, and there is a complaints of the necrosis also. Now, in our particular study, we have used a different strategy to see, because here we can see there is a kind of vascular changes that happens when there is a spinal cord. Now, there is a neurological deterioration. To address both the things, we want to see whether the transplantation of the mesenchymal stem cells and the combined treatment of vascular endothelial growth factor, that is VGF, which could able to produce the blood vessels, could able to rescue if we induce any spinal injury. So to address this, we have taken the mesenchymal stem cells from the dental tissue, dental pulp. So these are the specific morphology of the cells, how it looks like. And these are the cells which are stained with the nuclear stain, that is DAPI. And we have characterized these cells for the presence of mesenchymal population. And they are CD29 and they are CD90 positive cells, CD34 and CD45 negative cells. That indicates most of the cells that we have isolated from the dental pulp are mesenchymal in nature. And we wanted to see whether these mesenchymal stem cells have the differentiation ability to form other cell types. So we took these cells and we kind of given some differentiation factors to see whether they can form into different cell types. The same slide which I'm showing here, these cells could able to form the adipocytes, these cells could able to form the osteocytes, these cells could able to form the chondrocytes. That gave us the confidence that what are the cells we have isolated from the dental tissue could able to differentiate to the other cell types. And other thing I wanted to see whether these cell types could able to form the neuronal cell types also. So what we have done is we have taken the extracellular matrix from the brain, and then we have coated the plate with the extracellular matrix of the brain. On it, we have seeded, or you can say like we have plated the mesenchymal stem cells, and we have allowed for differentiation for 10 days. After 10 days, you can see the morphology from the fibroblastic in nature, they form like neurological in nature. That indicates like these mesenchymal stem cells, which are isolated from the dental pulp, have the ability to make neuronal cell type also. So that gave us the confidence like, okay, if we use the mesenchymal stem cells, these cells have the ability to form neuronal cell type also, because we are addressing now the spinal cord injury, which is a neuronal damage. Now, this is the laboratory setup. What we have here, we have a special machine called as infinite horizon impactor, wherein we create a spinal injury here. And then this is the place where we expose the spinal cord, and we expose the T8 to T line segments to expose the spine. And T9 to T10 thoracic spine is used for us to create the impact. And this is how the tissue looks like. This is the control tissue, and this is the injured tissue. You can see lot of cells that are coming. These are called as inflammatory cells, which are actually coming at the site of injury. Okay, and then here the tissue looks very, very neat and very healthy. But whereas when it is injured, the cell has other complications like degenerative changes. There's a proliferation of capillaries. There is a congested capillaries, and this indicates like whatever damage we have created, the damage is done. Okay, now this is our strategy to see it. How we have done the spinal cord injury. We have applied a force of 74 kilodynes for 30 seconds. And after that, after 3 days, we have given the vascular endothelial growth factor treatment of 606 micrograms per ml concentration. And after 3 days, we have given mesenchymal stem cells with ECM. So this is the strategy what we have adapted for the model that we have created. So I'm going to take through some of the videos which we have shown, which we have performed during the experimentation. The first video shows how we have created the injury. So this is considered the infinite horizon impactor, wherein we have uh, created the injury at the specific location on the thoracic segment of the spine. And this particular thing, yeah, this is the impact that we have created on the spinal cord for 30 seconds. Okay, and then after 30 seconds, this was removed. Okay, and later this animal looks like this. So because we had done it at thoracic segment, the hind limbs got compromised because the forelimbs are okay, but you can see there is a less movement of the hind limbs here. That indicates like we have created a very good model of the spinal cord. Now, to address this, there is a sham control. Sham control is nothing but we have opened the spinal cord, but we have not done any of the injury. That's why it is a control here. So it's a control where everything is normal. Now, this fourth slide shows you like has shown you what is this treatment strategy we have performed after this injury. After 3 days, we have introduced the vascular endothelial growth factor, and after 3 days, we have injected mesenchymal stem cells. And the readings that you see here at the fourth slide is an outcome of 60 days. After 60 days, what is the nature of the animal from the second slide? Here you can see the animal now looks pretty normal. The forelimbs are kicking nicely, indicating like there is a recovery of the spinal cord, and that spinal cord initially was been repaired, and then there is a free movement of the hind limbs. That indicates like whatever the treatment we have given, it has shown some impact of recovering the animal. So this is a small indication to show that the stem cells treatment has some beneficial impact on the nerve injuries. And there's another study which we are doing with LV Prasad Eye Hospital. This is regarding to the corneal blindness, which is done because of compromisation of the corneal epithelial cells. So as everybody knows, like the transparency of the cornea is very important for the optimal vision, and the epithelial cells on the cornea is important for corneal transparency. And this is been governed by the limbal stem cells that are present at the conjunctiva region. So if there is any changes in the limbal stem cells, it will ultimately reflect the corneal epithelial cells for the compromisation of the transparency. So any deficiency of the limbal stem cells could lead to a pathological condition called as limbal stem cell deficiency. And this is the main cause for loss of vision in large number of cases. So we want to address this issue, that how we could able to address if there is any vision loss due to limbal stem cell deficiency. Okay, now you can see it here. This is the cornea, and this is the limbus, and this is the conjunctiva. And this limbus contains the limbal stem cells, and these limbal stem cells contribute for the corneal epithelial cells to maintain the transparency. And if there is a damage at the limbus, it can lead to either partial limbal stem cell deficiency, or it can go for a complete limbal stem cell deficiency. And this limbal stem cell deficiency can occur due to chemical or alkali burns. When there is an alkali burn, like when some acid comes onto the contact with the eye completely, the acid or the alkali could able to burn the limbal cells or limbal stem cells, and this will lead to a condition called as pannus formation. And some of these patients are being treated with limbal stem cells, and after 2 years, you can see this eye is being completely recovered using this transplantation. So we want to create the same model in the rabbits to understand the pathology and the biology that will happen during the course of the process of recovering from the limbal stem cell deficiency. Okay, so this is how we do the limbal stem cell deficiency in the rabbit model. Now this whole indicates like the rabbits have been given some anesthesia, and after that we give alkali burn with sodium hydroxide, and we put some filter paper soaked in sodium hydroxide and keep it on the cornea, and it will we keep it for 1 minute, and then we'll take out the cornea, and then we will see with the fluorescent stain whether the corneal epithelial cells got removed or not. So this we see it with fluorescent staining, that we indicate like, okay, now the cornea is damaged. Now during the process, this is how it looked like. This is normal eye. After this eye has been kept for the alkali burn, after 15 days and after 45 days, this is how the cornea looks like. And we have a supporting document showing some of the inflammatory markers that will be there during this process. So this is how it looks like. And these are some of the histochemical parameters we actually look to check whether what kind of changes that happens after the post-alkali burn. And there will be a lot of epithelial hyperplasia, there will be surface ulceration, there will be goblet cells formation, there will be keratinization. So multiple factors are studied for this model system. And coming to this, now what we wanted to do is we wanted to replace the limbal stem cell deficiency with the healthy limbal stem cells. So what we do is we take a small portion of limbal tissue from the healthy eye, and we take out, we chop into small pieces, and we culture them in the laboratory. Okay, and then this is the explant of the limbal tissue. This from the limbal tissue, you can see a lot of cells that have been cultured or outgrowth from the tissue. This is the source of the limbal cells. Okay. Now, I will navigate you for the model what we created with this video. This is how we created the LSCD model. So this is how the rabbit will be given anesthesia. After giving the anesthesia, we put some surface paracrine so that there will be no pain on it. And later we will clean the cornea with the blade, so that some of the cells can be used for impression cytology experiments. So this paracrine is an anesthetic, so that there will be no pain on the animal. And then this particular filter paper will be soaked with sodium hydroxide and be kept on the surface. This is how the cornea looks like, and we give some anti-allergic drugs so that there will be no problem. And then later, so this is how the cornea looks like after the 2-3 months of the model. So what is the surgical treatment we have to do here? This is our laboratory. So this is the cornea, and we take small portion of the cornea, limbus tissue, and this limbus tissue will be taken in the petri dish. So from the healthy eye, we take a small portion of healthy limbal tissue, and we try to implant it on the animal which has a limbal stem cell deficiency. You can see the white eye, which has a pannus that is formed on the surface of the cornea. So first of all, what we do is we try to remove that pannus layer with the surgical blade, so that the cornea is exposed clearly. So that is the betadine to maintain antiseptic nature. And then, yeah, this is how we remove the pannus, so that the cornea will be little clear. And yeah, this is how we take out the pannus from the top. And then we put, what we do is we put some amniotic membrane on the top of this cornea. And then the biopsy that has been, the tissue that has been taken from the healthy part will be cut into small portions, and then we would like we will be keeping that cornea, that small pieces of the limbal tissue on the top of the amniotic membrane onto the diseased cornea, and then we try to keep them with the fibrin glue so that this cells, the membrane will not come out, and then this will be totally flushed with this normal saline, and then we stitch the eye. Okay, so this is how we have done the surgical this thing. So this is how we have done. So what we think is like the small portions or the bits of the limbal tissue that has been engrafted onto the amniotic membrane is placed on the diseased cornea, and these particular stem cells that are present on the surface could able to produce the new corneal epithelial cells on the surface, and this corneal epithelial cells will ultimately gives the transparency to the cornea to have a normal vision. So that is our anticipation, and still results are yet to come. The rabbits are under continuous observation, and then if any chance we want to present, we'll be presenting those data after we get them. So with this two things, I would like to conclude. Like first thing is like the MSCs and the vascular growth factor combination therapy had shown an improvement in the locomotor function following spinal cord injury, and the transplantation of the limbal stem cells in the LSCD model could be another indication of using the cells as one of the platforms to address the corneal blindness. So, so these are the two experimental projects which we are continuing in CCMB with the stem cells as one of the therapeutic option. So with this last slide, I thank you all very much for your patient understanding and patient listening to me, and I really appreciate if you have any things that is in specific to know about these studies. Thank you very much.
Thank you, sir. And it's the session of question and answer, sir. So can you please allow us? All the participants, please post your doubts, queries in the chat box. Yeah.
Sir, participants have posted questions in the chat box, sir. Shall we take up the questions? Sir, one of the participants asked that why would one use induced pluripotent stem cells when one could just use adult stem cells?
Yes, that is the whole idea of the pluripotency, just pluripotency, wherein you can take any cells from adult nature, and if you could able to induce that specific transcriptional factors, which I said you before, that KLF4, CMYC, SOX2, and NANOG, these particular transcription factors will govern that adult stem cells to pluripotent in nature.
Okay, sir. One more question. Can we correct genetic disorders like Down-Edward or any other which are prevalent in the society?
Yeah, investigations are going on as of now, but the real outcome is yet to come. People are using CRISPR-Cas9 mechanism to correct the genetic disorders and try to induce into the patients. Investigations are going on, but we need to give some time to get the fruitful outcome from these studies.
One more question, sir. Can they be used for siblings in case of emergency?
What is it?
Can they be used for siblings in case of emergency?
Siblings.
Yes.
So siblings. Yes, of course, because as mesenchymal stem cells, so these mesenchymal stem cells are have a little. There's you can say like there is something called as compatibility issues. So the compatibility issues should not be genetic in nature. So if there is any changes in between the siblings, then there cannot be an allogenic, or allogenic is okay, but from one patient to another patient, there are issues.
One more question, sir. Can stem cells be used to treat cancer patients?
Yeah, there is a new concept coming on called as CAR-T therapy, wherein you take the cells and then you decorate some trophic factors onto the cells, and then you target again this cells. So yes, there are very high profile studies going on using the CAR-T therapy. Yeah, it is there.
So one more question, which is better, induced pluripotent stem cells or embryonic stem cells?
IPSCs are the best because you are eliminating the issues of ethics, because you don't need to take it from the embryos, so you can generate your own cells from your own tissue. So IPSCs are more recommended than ESC cells.
Okay, sir, thank you. One more question, whom should the people approach for storage of stem cells? Whom should approach?
Yeah, it's a controversial question. People trying each and everything to store their cells. Yes, there are advantages, but the problem is when they use it. People store it, but who is going to take care of their what is I can say, like the storage facilities, and whether the cells are properly characterized, whether the cells have the same function as that were also isolated at the time of isolation. So there are multiple factors that will create a lot of problems in the future. So personally, I don't in a favor of preserving the cells.
Sir, is stem cells is used for autoimmune disorders?
Shravanti, unmute yourself, please.
Hello. Hello.
Shravanti, you are not audible. So, sir, any more questions?
Hello.
Okay, sir, thank you so much, sir.
Yeah, it's a great privilege coming to your college virtually.
Fortunate to have you.
Virtually, so thank you all very much for your kindness gesture and giving me time to share some of our laboratory findings also.
Yes.
I wish I could come there, but because of the pandemic, we could not. But sometime
We will definitely invite you, sir, to our institutions. Definitely, our students will inspire by you and they learn something about this stem cell research. So we'll plan
Hello.
for that in the future.
Sorry, sir. Hello.
Yeah, you are audible.
Yeah. Thank you, sir. I have one more doubt, sir.
yeah.
Is this stem cell therapy is useful for autoimmune disorders?
That's what there is a concept going on called as CRISPR-Cas9 mechanism.
Okay.
So this is this is editing of the cells using genetic tools. So they kind of correct some of the cells and then again reintroduce into the body, so that that particular cells will again repair the cells which are present in the body. So they are in the preclinical stage, but we can soon hear that it can be also used for not only autoimmune but many other disorders as well.
Okay, sir. Thank you so much for
Thank you.
clarifying our doubts, sir. Now I would like to call upon my colleague to present the vote of thanks. Swati, over to you.
Thank you, Shravanti. Good afternoon once again. On behalf of Telangana Social Welfare Residential Degree College for Women, Jagadgiri Gupta, I'm here to present the vote of thanks to everyone. So first of all, I would like to express my sincere gratitude to today's speaker of this webinar, Dr. P. Kiran Kumar, for accepting our invitation. Thank you, sir, for taking out time from your busy schedule. And you enlightening us with valuable research work, especially on stem cells, embryonal stem cells especially, and their potential to differentiate into different cells and the same cells, and the use of the stem cells in pathology in curing disorders. So in the last, we felt that, especially in the last, we were in the lab, we felt that. So that you have taken us into the lab. And today's webinar was full of knowledge and more interesting things. It gave deep insights into the topic and also revealed some interesting facts. So that point where you told us about the stem cells was really informative, sir. And I'm pretty much sure the precious knowledge which was given by you definitely help in further studies or in research of the students and teachers as well. So thank you so much, sir, for being with us in this today's webinar. Thank you very much. Now I would like to express my gratitude to Dr. R.S. Praveen Kumar, IPS Secretary, TSW REIS and TTW REIS for giving numerous opportunities to the students and to the faculty as well in the organization. Thank you, sir. I also extend my thanks to Dr. Praveen Mamidala, sir, Joint Secretary and Head of Higher Education Department, TSW REIS, for his constant support and for motivating us to do various activities. Thank you, sir. I thank Dr. P. Bhanuprasad, sir, OSD, TSW RDCs for encouraging towards different activities. Thank you, sir. And I always wish to thanks to Dr. P. Rajni ma'am, Zonal Coordinator, TSW RDC South Zone, for encouraging us to do various activities in the institutions. Thank you, ma'am. I would also like to thank our principal, Himabindu ma'am, for giving permission to organizing this webinar session and giving encouragement, constant support in conduction of today's webinar. Thank you, ma'am. I also thank our Vice Principal B. Nutana ma'am and my colleagues for giving advices. And I also express my special thanks to Department of Computer Science and Librarian G. Lakshmi Prasanna for giving technical support. And last but not the least, I thank all the participants for attending the webinar and making it successful. So thank you so much. With these words, I would like to end this session. Stay safe, stay home. Thank you all.
Thank you. Thank you. Thank you.
Thank you. Thank you so much, sir.
Thank you, sir.
you.
Thank you. Thank you.